Settle into the Sleepy Mind channel. Tonight, we explore herbal remedies. If you enjoy stories like these, like and subscribe for more. New videos are posted weekly. Let us begin. Long before laboratories measured chemical reactions, humans identified plants that slowed heart rate, reduced pain signals, and limited infection in damaged tissue. These effects were observed repeatedly and preserved because they produced consistent and measurable changes in human physiology. Early knowledge formed through direct interaction with plants, where survival depended on recognizing which compounds produced reliable biological results. These observations did not rely on theory or written explanation. They were based on direct cause and effect, where a plant was used and a physical response followed. When a plant reduced swelling or eased pain, that result could be seen and felt in a clear and immediate way. Over time, patterns formed, and certain plants became associated with specific outcomes because those outcomes could be repeated across different situations. Certain plants contain compounds that bind to receptors in the brain, altering neural signaling linked to stress and relaxation responses. These interactions change how signals are transmitted between nerve cells, reducing the intensity of stress-related activity. As a result, a measurable shift in how the body responds to tension, often observed through slower breathing, reduced muscle tightness, and a decrease in overall alertness. Other compounds influence enzyme activity in the digestive system, accelerating food breakdown and reducing discomfort in the stomach. These compounds increase the efficiency of chemical processes that convert food into usable nutrients. As digestion becomes more efficient, pressure within the digestive tract decreases, and the likelihood of discomfort is reduced. This effect can be observed through smoother digestion and a reduction in bloating or irritation. Some roots and bark contain molecules that disrupt microbial growth by damaging bacterial cell structures. These compounds interfere with the integrity of microbial cells, preventing them from functioning normally. As a result, the spread of harmful bacteria is limited, allowing the body to maintain a more stable internal environment. This effect was often observed through reduced infection and faster recovery in damaged tissue. A compound found in willow bark reduces inflammation by blocking chemical signals that trigger swelling and pain in tissue. The process limits the body's inflammatory response, preventing excessive swelling that can interfere with normal movement. The reduction in these signals allows tissue to recover more efficiently while maintaining structural stability. Alkaloids from the opium poppy bind to receptors in the nervous system, reducing the transmission of pain signals along neural pathways. These compounds alter how pain is perceived by changing the way signals are processed within the brain. As a result, a noticeable decrease in discomfort, even when the source of the pain remains present. These interactions occur at a measurable level, where plant compounds directly influence biological function through defined mechanisms. Every compound produces a specific effect based on how it interacts with receptors, enzymes, and cellular pathways. The relationship allows plant-based effects to be observed, repeated, and relied upon when the same conditions are present. Across forests and grasslands, plants produce chemical defenses that protect against insects, fungi, and bacterial invasion. These compounds are part of the plant's survival system, allowing it to resist damage and maintain growth in a competitive environment. When these compounds are introduced into the human body, they continue to perform similar functions by interacting with biological systems. When consumed or applied, these compounds interact with human physiology and produce effects such as increased circulation and reduced inflammation. These responses occur because the compounds influence how blood flows through tissue and how the body regulates its response to stress and injury. As a result, a measurable improvement in how the body maintains stability under changing conditions. The relationship between plant survival chemistry and human biological response forms the foundation of herbal medicine. What begins as a defense mechanism within the plant becomes a functional interaction within the human body. The same compounds that protect the plant can support regulation and recovery when introduced into human systems. Over time, this connection became more clearly understood through repeated use and observation. Plants were not selected randomly, but chosen because they produced reliable and consistent outcomes. Each successful use reinforced the connection between a specific plant and a specific physiological effect. The process created a system where plant-based compounds could be used with increasing precision. As more observations were collected, patterns became clearer, and the use of plants became more structured. This allowed herbal practice to develop into a method based on measurable results rather than assumption. The consistency of these results depended on the ability to recognize subtle differences in plant effects. Some plants produced strong and immediate responses, while others produced gradual changes that became noticeable over time. Understanding these differences allowed for more accurate selection based on the desired outcome. In this way, early herbal knowledge developed as a direct extension of biological observation. Each plant represented a chemical system capable of interacting with the body in a predictable way. These interactions form the basis for understanding how natural compounds could influence human physiology through direct and measurable effects. As these observations accumulated, they established a reliable framework that connected plant chemistry with human response. This framework did not rely on abstract theory, but on repeated evidence that could be observed and confirmed. The result was a practical system grounded in consistent biological interaction, forming the starting point for the continued development of herbal medicine. Preparation methods determine how these compounds enter the body and how efficiently they produce measurable physiological effects. The way a plant is processed changes how its chemical components are released, absorbed, and used by biological systems. This means that the same plant can produce different results depending on how it is prepared before use. Water-based preparations release soluble compounds that move through digestion and enter the bloodstream through intestinal absorption. When plant material is exposed to water, compounds that dissolve easily begin to separate from the plant structure. Heat increases this process by causing molecules to move more rapidly, allowing a greater number of compounds to be released into the liquid. This forms a preparation that can be absorbed steadily as it passes through the digestive system. Once absorbed, these compounds travel through circulation and reach target tissues where they produce their effect. The speed and strength of this effect depend on how quickly the compounds move through digestion and how efficiently they are transferred into the bloodstream. The process creates a gradual onset that produces a steady and measurable change within the body. Alcohol-based preparations preserve more complex molecules, maintaining stability and increasing concentration for controlled use. Certain compounds do not dissolve easily in water and require a different medium to be extracted effectively. Alcohol allows these compounds to remain intact while also preventing rapid breakdown over time. This results in a preparation that contains a wider range of active components in a more concentrated form. Because of this increased concentration, smaller amounts can produce noticeable effects within the body. The stability of these compounds also allows the preparation to maintain its effectiveness over longer periods. This makes alcohol-based methods useful when consistent potency is required across repeated use. As knowledge developed, cultures organized plant use into systems based on observable effects within the body. These systems were not based on assumption, but on repeated observation of how specific preparations produced consistent outcomes. Over time, this led to the formation of structured approaches where preparation methods and plant selection were combined to achieve predictable results. Some traditions combined multiple plants to influence several biological pathways and produce coordinated physiological responses. Each plant contributed a specific effect, and when used together, these effects worked in combination to support multiple systems at once. This allowed for a broader response within the body, without relying on a single compound to produce all results. The combination of plants required careful observation to ensure that each component contributed to the intended outcome. If one plant produced a strong effect, while another produced a milder response, the balance between them had to be adjusted to maintain consistency. The process created a more refined approach, where plant combinations were selected based on how they interacted with each other, as well as with the body. Other approaches focused on individual plants, selected for specific outcomes such as improved circulation or reduced inflammation. In these cases, a single plant was chosen because it produced a clear and reliable effect on a particular system. This method allowed for more direct observation of how one plant influenced the body without the complexity of combined interactions. The choice between using single plants or combinations depended on the desired outcome and the condition being addressed. Some situations required a focused effect, while others benefited from a broader response involving multiple systems. This flexibility allowed herbal practice to adapt to different needs while maintaining consistency in results. Repeated use revealed consistent patterns, linking plant chemistry with measurable results in human physiology. As the same preparations were used over time, the effects they produced became more predictable. This allowed for greater confidence in selecting specific plants and preparation methods based on the expected outcome. Bitter compounds stimulated digestive secretions and improved nutrient breakdown within the digestive tract. These compounds activated receptors that increased the release of digestive enzymes, allowing food to be processed more efficiently. The result was a smoother digestive process with reduced buildup and discomfort. Aromatic compounds influenced neural pathways, altering heart rate and modifying stress-related responses. These compounds interacted with sensory receptors linked to smell, which are directly connected to areas of the brain that regulate emotion and physiological response. This created a rapid effect where changes in scent could influence relaxation and reduce physical tension. These patterns were not random, but consistent across different individuals and conditions when preparation remained the same. This consistency allowed herbal practice to move beyond trial and error into a more structured system where outcomes could be predicted with greater accuracy. The effectiveness of these preparations depended on maintaining consistency in both plant selection and method of use. Small changes in preparation could alter how compounds were released and how strongly they interacted with the body. This meant that careful attention to detail was necessary to ensure that results remained stable across repeated use. As these systems developed, preparation methods became an essential part of how plant-based compounds were used. The plant itself was only one part of the process, while the method of extraction determined how those compounds would behave within the body. This connection between preparation and outcome became a defining feature of herbal practice. Over time, this understanding created a reliable framework where plant compounds could be used in a controlled and repeatable way. Each preparation method produced a specific type of interaction, and each interaction could be observed through measurable changes in biological function. This allowed herbal medicine to develop as a system grounded in consistency, where both the plant and its preparation played a role in producing predictable physiological effects. Modern analysis has isolated many plant compounds and identified their structure at a molecular level. The process allows scientists to examine how individual compounds interact with specific biological systems through direct chemical pathways. By separating these compounds from the plant, it becomes possible to observe their effects with greater precision and consistency. Every compound has a defined structure that determines how it behaves when introduced into the body. This structure influences how the compound binds to receptors, interacts with enzymes, and moves through cellular pathways. These interactions are not random, but follow predictable patterns based on chemical compatibility between the compound and biological systems. The process connects traditional knowledge with measurable biochemical activity and links observed outcomes with defined mechanisms. What was once understood through repeated observation can now be explained through detailed analysis of how compounds function at a molecular level. This connection provides a clearer understanding of why certain plants produce consistent effects across different individuals. When a compound interacts with a receptor, it changes how signals are transmitted within the body. The interaction can increase or decrease activity depending on how the compound influences the receptor's function. As a result, a measurable change in processes such as pain perception, inflammation, or neural activity. Many pharmaceutical drugs still rely on plant-derived compounds used directly or reproduced through synthesis. In some cases, the original compound is extracted and used in its natural form. In other cases, the structure of the compound is replicated or modified to produce a similar effect with controlled precision. The relationship shows that plant-based compounds and modern pharmaceuticals are connected through shared chemical foundations. The difference lies in how the compounds are refined, concentrated, and delivered within the body. While pharmaceuticals often isolate a single compound, plant-based preparations may contain multiple compounds that work together to produce a combined effect. Today, plant-based compounds remain widely used to support digestion, circulation, immune response, and cellular repair. These compounds interact with systems that regulate essential functions, allowing the body to maintain stability under changing conditions. Their continued use reflects the consistency of their effects when applied correctly. The effectiveness of these compounds depends on concentration, preparation, and interaction with biological systems that determine the final outcome. A compound must be present in sufficient quantity to produce a measurable effect, while also being delivered in a way that allows it to reach its target within the body. These factors influence how quickly and strongly the compound produces its response. Each plant represents a chemical system shaped by environmental pressure, nutrient availability, and biological competition. These conditions influence the type and concentration of compounds produced within the plant. As a result, the same plant species can vary in potency, depending on where and how it is grown. This variability requires careful selection to ensure that plant material produces consistent results. Factors such as soil composition, sunlight exposure, and water availability all affect how compounds develop within plant tissues. Understanding these influences allows for more accurate selection of plant material based on its expected effect. Ancient medical systems developed around these observations, organizing plant use into structured practices based on consistent physical outcomes. These systems were built on repeated experience, where the same plant preparations produce the same results when used under similar conditions. This allowed knowledge to be refined over time through practical application. In ancient Egypt, written records describe plant preparations used to treat wounds, infections, and digestive disorders with measurable effects. These records show that plant-based treatments were selected based on observed results, rather than assumption. Each preparation was documented because it produced a consistent change in the body. These texts also reflect an early understanding of how preparation influenced effectiveness. Different methods were used depending on the type of plant and the condition being addressed. This demonstrates that even early systems recognize the importance of both plant selection and preparation in producing reliable outcomes. As knowledge continued to develop, these structured systems allowed plant use to become more consistent across different regions and conditions. The same plant could be used in different environments, with similar results when preparation remained controlled. This consistency reinforced confidence in plant-based practices and supported their continued use. The connection between traditional systems and modern analysis highlights a continuous line of observation and refinement. While the tools used to study plant compounds have changed, the underlying principle remains the same. Plant compounds produce measurable effects that can be observed, repeated, and explained through both practical experience and scientific analysis. This continuity allows plant-based knowledge to remain relevant in modern contexts, where traditional practices can be understood through detailed examination of chemical structure and biological interaction. As a result, a clearer understanding of how plant compounds influence the body through defined and measurable mechanisms. As this understanding expands, it provides a framework for selecting and using plant-based compounds with greater precision. Every compound can be evaluated based on how it interacts with specific systems, allowing for more controlled and predictable outcomes. The process strengthens the connection between observation and explanation, ensuring that plant-based practices remain grounded in measurable biological effects. In ancient China, herbal practice developed into a system that combined multiple plants to influence different functions within the body. This system was built on repeated observation, where specific combinations produce consistent and measurable effects across different individuals. Each plant was selected based on how it contributed to the overall response, allowing multiple biological systems to be influenced at the same time. These combinations were designed to produce coordinated effects, such as improving circulation while reducing inflammation within tissues. One plant might increase blood flow, while another limited inflammatory signaling, creating a balanced response that supported recovery without overstimulating the body. This approach allowed for more controlled outcomes by distributing the effect across multiple pathways rather than relying on a single compound. The selection of each plant was based on observed results that could be repeated across different individuals. If a combination produced the same outcome under similar conditions, it became part of established practice. This repetition allowed practitioners to refine combinations over time, improving consistency and predictability in how the body responded. In India, Ayurvedic practice classified plants according to their influence on digestion, energy use, and internal balance within the body. This system focused on how plant compounds interacted with core functions that regulate stability and adaptation. Each plant was assigned a role based on its measurable effect on processes such as nutrient absorption, metabolic activity, and recovery from physical strain. Plants were selected to produce specific outcomes, such as improving nutrient absorption or supporting recovery from physical strain. Some compounds enhance digestive efficiency, allowing nutrients to be broken down and absorbed more effectively. Others supported energy use by influencing how the body processed and distributed those nutrients. This created a system where plant use was directly connected to maintaining functional balance. This system relied on direct observation of how plant compounds affected measurable biological functions. Effects were not assumed, but confirmed through repeated use, where changes in digestion, energy levels, and recovery could be clearly identified. Over time, this allowed for a structured approach where plants were chosen based on how they influence specific processes within the body. Across Europe, early practitioners used local plants to treat common conditions through methods that focused on consistent results within the body. These practices were shaped by the availability of regional plant species and the need to address conditions such as infection, inflammation, and digestive discomfort. Each plant was selected based on its ability to produce a reliable and observable effect. Willow bark was used to reduce pain and swelling through compounds that block inflammatory signaling. These compounds limited the release of molecules that trigger swelling, allowing tissue to recover without excessive inflammation. This effect could be observed through reduced discomfort and improved movement in affected areas. Garlic was used to limit infection by introducing compounds that disrupt microbial growth within tissue. These compounds interfered with the structure and function of bacterial cells, reducing their ability to multiply and spread. The result was a measurable reduction in infection and a more stable internal environment. These European practices emphasized practical application, where results were evaluated based on physical response rather than theoretical explanation. Plants that consistently produced beneficial outcomes were retained, while those that did not were excluded from continued use. The process created a refined selection of plants that could be relied upon for specific effects. Although these systems developed in different regions, they shared a common foundation based on observation, repetition, and measurable outcomes. Each system identified plants that produced consistent effects and organized their use into structured methods. The differences between them reflected regional variation, while the underlying principle remained the same. This principle was based on the idea that plant compounds interact with biological systems in predictable ways. When the same plant was used under similar conditions, it produced similar results. This allowed knowledge to be transferred and adapted across different environments while maintaining consistency in outcomes. The development of these systems also required an understanding of how plants interacted with each other when used in combination. Some combinations enhanced the overall effect, while others reduced it or created imbalance. Careful observation allowed practitioners to identify which combinations produced the most stable and effective results. As these practices continued to evolve, they became more structured and refined, with clear relationships between plant selection, preparation, and outcome. Each step in the process contributed to the final effect, creating a system where results could be predicted based on known interactions between plant compounds and biological systems. The consistency of these systems reflects the stability of the relationship between plant chemistry and human physiology. Despite differences in cultural approach, the same types of compounds produce similar effects across different populations. This consistency reinforced the reliability of plant-based practices and supported their continued development. Over time, these regional systems contributed to a broader understanding of how plant compounds could be used to influence the body in controlled and predictable ways. Each system added to the collective knowledge by identifying new plants, refining preparation methods, and improving the accuracy of plant selection based on observed outcomes. This accumulation of knowledge created a foundation that allowed plant-based practices to expand beyond local use and become part of a wider framework of understanding. The shared reliance on measurable effects ensured that these practices remained grounded in observable biological response rather than assumption. As a result, the development of herbal systems across different regions represents a continuous process of observation, refinement, and application. Each system reflects a structured approach to using plant compounds in a way that produces consistent and reliable effects within the body. This structure forms an essential part of how herbal medicine developed into a practice based on measurable interaction between plant chemistry and human physiology. In the Americas, indigenous communities identified hundreds of plant species that produced reliable effects when applied to wounds or consumed for internal conditions. These plants were selected through repeated observation, where their effects could be seen in recovery, resistance to infection, and improvement in physical function. Each plant was tested through use over time, and only those that produced consistent results became part of established practice. Plants such as echinacea were used to support immune response by increasing activity within defense systems. The compounds in echinacea interact with immune cells, enhancing their ability to recognize and respond to foreign organisms. The interaction improves the speed and efficiency of the body's response during the early stages of infection, reducing the likelihood of widespread illness. These practices were based on repeated observation of outcomes that could be measured through recovery and resistance to illness. When a plant consistently reduced infection or supported healing, it became associated with that specific outcome. Over time, this created a system where plants were selected based on how reliably they influenced biological processes within the body. The use of plants in these communities extended beyond isolated treatment and became part of a broader understanding of how the body responds to environmental conditions. Plants were used not only to address immediate issues, but also to support ongoing stability within the body. This approach recognized that maintaining balance could reduce the likelihood of more serious conditions developing. The transmission of this knowledge depended on careful observation and repetition across generations. Information was passed through direct teaching, where each preparation method was demonstrated and refined through practical use. This ensured that the knowledge remained accurate and that preparation methods were applied consistently to produce the same results. Each generation contributed to refining this knowledge by observing how plants behaved under different conditions. If a plant produced slightly different effects depending on preparation or timing, those differences were noted and incorporated into practice. The process allowed knowledge to evolve while maintaining its foundation in measurable results. The reliance on direct teaching ensured that subtle details were preserved, including how plants were selected, prepared, and applied. These details often determined the effectiveness of the plant, as small changes in preparation could alter how compounds were released and how they interacted with the body. This level of precision allowed for consistent outcomes across repeated use. As trade routes expanded, plant knowledge moved between regions, introducing new species and new applications into existing systems. This exchange allowed communities to observe how unfamiliar plants produced effects similar to those already known. When a new plant produced a comparable response, it could be integrated into existing practices with minimal adjustment. Spices such as cinnamon and ginger were exchanged and adopted for their ability to influence digestion and circulation through identifiable chemical compounds. Cinnamon was observed to improve circulation by influencing blood flow, while ginger supported digestion by increasing movement within the digestive system. These effects were consistent enough to allow these plants to become widely used across different regions. The introduction of new plants expanded the range of available treatments, allowing for greater flexibility in how conditions were addressed. If one plant was unavailable, another with a similar effect could be used as a substitute. This adaptability strengthened the overall system by reducing dependence on a limited number of plant species. The exchange of plant knowledge also allowed for comparison between different methods of preparation and use. If one culture prepared a plant differently but achieved the same result, that method could be evaluated and potentially adopted. The process improved the efficiency and consistency of plant-based practices by identifying the most effective approaches. As knowledge spread, it became clear that the effectiveness of plant compounds depended not only on the plant itself, but also on how it was prepared and used. This reinforced the importance of maintaining consistency in preparation methods to ensure predictable outcomes. Each step, from harvesting to application, influenced the final effect produced within the body. The integration of new plants into existing systems required careful observation to ensure that their effects remained consistent across different individuals and conditions. The process ensured that only plants with reliable and measurable effects were retained. Over time, this created a more comprehensive system that combined knowledge from multiple regions. The expansion of plant knowledge through trade and exchange demonstrates how observation and adaptation contributed to the development of herbal practice. Each new plant added to the system was evaluated based on its ability to produce consistent outcomes. This maintained the integrity of the practice while allowing it to grow and evolve. The continued use of these plants reflects the stability of their effects across different environments and populations. Despite variations in climate and preparation, the same compounds produced similar responses within the body. This consistency reinforced confidence in plant-based practices and supported their continued use across generations. The combination of observation, transmission, and exchange created a system where plant knowledge could expand while remaining grounded in measurable results. Each new addition to the system was evaluated through direct interaction with the body, ensuring that only effective plants became part of established practice. The process allowed herbal medicine to develop as a reliable and adaptable system based on consistent biological response. The preparation of these plants followed methods that preserved their active compounds and ensured consistent results. Each step in the preparation process influenced how the compounds remained stable and how effectively they could be used by the body. Without proper preparation, many compounds would break down before they could produce a measurable effect. Drying plant material reduced moisture and prevented degradation of chemical structures that produced biological effects. When moisture remains in plant tissue, it creates conditions where compounds can break down more rapidly through chemical and microbial processes. By removing this moisture, the internal structure of the plant stabilizes, allowing the compounds to remain intact over longer periods. This preservation method also allows plant material to be stored and used when needed without losing effectiveness. Once dried, plant material can be transported, stored, and prepared at a later time, while still maintaining its chemical profile. This makes drying an essential step in maintaining consistency across repeated use. Grinding and extraction methods increase the availability of compounds during use, improving their interaction with the body. When plant material is broken down into smaller particles, more surface area is exposed, allowing compounds to be released more efficiently during preparation. This increases the likelihood that a sufficient amount of active compounds will enter the body to produce a measurable effect. Extraction methods further enhance this process by separating compounds from plant tissue into a medium that can be easily absorbed. Whether through water or alcohol, extraction allows compounds to move from a solid structure into a form that interacts more directly with biological systems. This step ensures that the compounds are available in a usable form rather than remaining trapped within plant fibers. The effectiveness of these preparations depended on maintaining consistency in both plant selection and method of use. If preparation varied, the concentration of active compounds could change, leading to differences in how strongly the plant affected the body. This required careful attention to ensure that each preparation produced a similar result when used under the same conditions. Even small changes in preparation could alter how compounds were released and how they behaved within the body. A shorter extraction time might result in a weaker preparation, while longer exposure could increase concentration beyond the intended level. This sensitivity to preparation highlights the importance of precision in achieving consistent outcomes. Over time, these practices formed a foundation that connected plant-based knowledge across cultures through shared observation of biological response. Although different regions use different plants, the underlying methods of preparation followed similar principles. These principles were based on preserving compounds, increasing availability, and ensuring that the body could absorb them effectively. This shared approach allowed knowledge to transfer between cultures, even when the specific plants differed. If a preparation method proved effective in one region, it could be applied to different plant species in another region with similar results. This created a common framework that supported the expansion of plant-based practices. The connection between preparation and outcome became a defining feature of herbal practice. A plant was not considered effective simply because of its natural properties, but because of how those properties were preserved and delivered. This understanding ensured that preparation remained an essential part of producing reliable results. As these methods were refined, they became more consistent and structured, allowing practitioners to predict how a preparation would behave before it was used. This predictability reduced uncertainty and increased confidence in plant-based treatments. Each step in the process contributed to a controlled interaction between plant compounds and the body. The ability to maintain this level of consistency depended on recognizing the relationship between plant structure and chemical behavior. Different parts of a plant, such as leaves, roots, or bark, required different preparation methods to release their compounds effectively. Understanding these differences allowed for more accurate preparation and improved outcomes. For example, softer plant materials such as leaves release their compounds more quickly and require less processing. In contrast, denser materials such as roots and bark require longer preparation to break down their structure and release their compounds. Adjusting preparation methods based on these characteristics ensures that compounds are extracted efficiently without being degraded. The temperature used during preparation also influences how compounds behave. Higher temperatures increase molecular movement, allowing compounds to separate more quickly from plant tissue. However, excessive heat can also break down certain compounds, reducing their effectiveness. Maintaining the correct temperature range is essential for preserving the balance between extraction and stability. Time plays a similar role in determining the strength and effectiveness of a preparation. Short preparation times may not allow enough compounds to be released, while extended times can lead to excessive concentration or degradation. Finding the correct balance ensures that the preparation produces a consistent and measurable effect. The interaction between these factors creates a controlled system where preparation determines how plant compounds will behave once introduced into the body. Each variable, including moisture, particle size, temperature, and time, contributes to the final outcome. By managing these variables carefully, consistent results can be achieved across repeated use. As this understanding developed, preparation methods became standardized within different practices. This standardization allowed for greater consistency and made it easier to reproduce results under similar conditions. It also allowed knowledge to be shared more effectively, as preparation methods could be taught and repeated with predictable outcomes. The refinement of these methods reflects the importance of preparation in determining the effectiveness of plant-based compounds. While the plant provides the raw material, preparation transforms that material into a form that can interact with the body in a controlled and measurable way. This transformation is essential for producing reliable results. In this way, preparation acts as the link between plant chemistry and biological response. Without proper preparation, the compounds may remain inactive or unavailable, limiting their ability to influence the body. With correct preparation, those same compounds become accessible and capable of producing measurable physiological effects. The relationship ensures that preparation remains a central part of herbal practice, where each step contributes to the final outcome. By maintaining consistency in preparation methods, plant-based compounds can be used with confidence, producing predictable effects that support stability and function within the body. As these practices continued to develop, the relationship between plant compounds and human biology became more consistent through repeated observation. Each use provided additional confirmation that specific compounds produce specific effects under the same conditions. This allowed plant-based knowledge to move from isolated experience into a more structured and reliable system. Specific plants were selected not because of belief, but because they produced clear outcomes such as reduced swelling, controlled pain, and improved recovery from injury. These outcomes could be observed directly, allowing practitioners to associate each plant with a measurable biological response. Over time, this created a clear link between plant selection and expected results. This consistency allowed plant use to become more structured, with preparation methods refined to preserve the compounds responsible for these effects. As the connection between compound and outcome became clearer, less variation was tolerated in how plants were prepared and applied. This ensured that the same plant would produce the same result when used under similar conditions. In many regions, combinations of plants were used to influence multiple responses within the body at the same time. This approach allowed different systems to be supported simultaneously, creating a more balanced and coordinated effect. One plant might improve digestion, while another reduced inflammation, allowing the body to respond more efficiently to physical stress. The use of combinations required careful observation to ensure that each plant contributed to the intended outcome. If two plants influenced the same pathway too strongly, the combined effect could become excessive. If they worked in different ways, they could complement each other and produce a more stable response. This balance was essential for maintaining predictable results. These combinations were adjusted based on observation, ensuring that each component contributed to a defined outcome. When a combination produced consistent results, it was repeated and refined until it became part of established practice. The process allowed combinations to evolve into reliable systems that could be used with confidence. The reliability of these results depended on recognizing that not all plants produce the same effect in every condition. A plant that reduced inflammation in one situation might produce a different response under different circumstances. This required careful selection based on the specific condition being addressed. Selection required attention to the specific outcome being observed, whether that was reduced pain, improved circulation, or faster tissue repair. Each plant had to be matched to the condition based on how its compounds interacted with biological systems. This level of precision ensured that plant use remained connected to measurable results rather than assumption. As knowledge expanded, certain plants became widely recognized for their consistent effects across different regions and cultures. These plants were used repeatedly because they produced reliable changes in key systems such as inflammation, digestion, and immune response. Their consistent performance reinforced their value within herbal practice. Willow bark, garlic, ginger, and turmeric were used repeatedly because they produced reliable changes in inflammation, digestion, and immune response. These plants became central to many practices because their effects could be observed and confirmed across different individuals and conditions. This repetition strengthened confidence in their use and supported their continued application. These repeated outcomes strengthened confidence in plant-based practices and supported their continued use over time. As more observations confirmed the same effects, the relationship between plant compounds and biological response became more clearly defined. This allowed plant-based systems to become more structured and predictable. The continued use of these plants reflects a stable relationship between plant chemistry and human biological response. Every compound produces a specific effect based on how it interacts with receptors, enzymes, and cellular systems within the body. The interaction remains consistent when preparation and dosage are controlled. Every compound produces a specific effect based on how it interacts with receptors, enzymes, and cellular systems within the body. These interactions create measurable changes that can be observed through physical response. This enables plant-based compounds to be used with greater accuracy when the underlying mechanisms are understood. These effects can be observed, repeated, and relied upon when preparation and selection remain consistent. This reliability forms the foundation of herbal medicine, where plant compounds continue to influence human health through direct and measurable interaction. The stability of this relationship ensures that plant-based practices remain grounded in consistent biological response. As this system became more refined, it allowed practitioners to anticipate how the body would respond before a plant was used. This predictive ability reduced uncertainty and improved the effectiveness of plant-based treatments. Each decision, from plant selection to preparation, became part of a structured process designed to produce a specific outcome. This level of refinement marks the transition from simple observation to controlled application. What began as individual experience developed into a system where plant compounds could be used in a consistent and predictable way. This structure continues to define how herbal medicine operates, connecting plant chemistry with measurable physiological effects through careful selection and application. Plants used in herbal practice are selected based on the specific compounds they contain and the measurable effects those compounds produce within the body. Each plant contains a unique combination of chemical structures that determine how it interacts with biological systems. These compounds influence processes such as inflammation, digestion, circulation, and neural activity through direct interaction with receptors and cellular pathways. This selection process is not based on appearance or general classification, but on how a plant consistently affects the body when used under controlled conditions. When a plant produces the same measurable effect across repeated use, it becomes associated with that specific outcome. This enables practitioners to choose plants based on predictable biological responses rather than assumption. Each plant contains a chemical profile that defines its function within the body. This profile includes multiple compounds that may act on different systems at the same time. Some compounds produce a primary effect, while others support or modify that effect, creating a balanced interaction within the body. This combination allows a single plant to influence more than one process simultaneously. Chamomile contains apigenin, a compound that binds to receptors in the brain and reduces neural activity associated with stress response. The interaction decreases signaling within pathways that regulate alertness and tension. As a result, a measurable reduction in physical restlessness, slower breathing, and a more stable transition into a relaxed state. Peppermint contains menthol, which relaxes smooth muscle tissue within the digestive tract and improves the movement of food through the stomach. This action reduces pressure caused by gas buildup and delayed digestion. As a result, improved digestive flow, allowing the body to process food more efficiently and reducing discomfort associated with slowed movement. Ginger contains compounds that stimulate gastric emptying, increasing the rate at which food moves from the stomach into the small intestine. This effect reduces nausea by preventing the accumulation of partially digested material. The outcome is a noticeable reduction in discomfort linked to motion, imbalance, and digestive disruption. Turmeric contains curcumin, a compound that reduces inflammatory signaling by limiting the production of molecules that trigger swelling. The interaction supports tissue recovery after strain or minor injury by reducing excess inflammation. The effect can be measured through improved movement, reduced stiffness, and decreased irritation in affected areas. Garlic contains sulfur-based compounds that increase immune activity by enhancing the responsiveness of white blood cells. These compounds interfere with microbial growth and support the body's ability to respond to infection. As a result, an increase in defense activity that can be observed through improved resistance to bacterial presence. Echinacea contains active compounds that stimulate immune response by increasing activity in defense-related cells. This stimulation improves the body's ability to identify and respond to foreign organisms during the early stages of infection. The outcome is a faster and more efficient immune reaction that supports recovery. Lavender produces aromatic compounds that influence neural pathways through receptors linked to smell. These compounds reduce activity in regions of the brain associated with stress response, creating a calming effect. This effect can be observed through reduced heart rate, lower muscle tension, and a shift toward a more relaxed physiological state. Valerian root influences neurotransmitter levels within the brain, increasing signals associated with relaxation and sleep readiness. The interaction supports the transition from wakefulness to sleep by reducing neural activity linked to alertness. As a result, improved sleep onset and a more stable transition into rest. Aloe vera contains compounds that support skin repair by maintaining moisture and promoting cellular regeneration within damaged tissue. When applied to the skin, these compounds create an environment that supports healing while reducing irritation. The effect can be observed through faster recovery and reduced surface inflammation. Rosemary contains compounds that stimulate circulation by increasing blood flow to targeted regions within the body. This increased flow improves oxygen delivery and nutrient transport within tissue. The outcome is improved responsiveness within the nervous system and enhanced support for cognitive function. Different plants produce different types of effects depending on how their compounds interact with specific systems within the body. Some act quickly and produce noticeable changes within a short period, while others require repeated use to build consistent results over time. The timing and duration of these effects depend on how the compounds are absorbed, distributed, and processed within the body. The consistency of these interactions allows plant compounds to be used in a structured and repeatable way when preparation and dosage remain controlled. Every compound produces a defined effect based on its chemical structure and biological compatibility. The relationship allows practitioners to match specific plants to specific outcomes with increasing accuracy. The selection of plants based on their chemical profiles creates a system where herbal practice can operate with precision rather than generalization. Each plant is chosen because it produces a measurable effect, and that effect can be observed and confirmed through repeated use. This supports that plant-based compounds are used in a way that remains consistent with their biological function. Over time, this approach has allowed herbal practice to become more refined, with clear relationships between plant compounds and physiological response. Each plant contributes to this system by providing compounds that interact with the body in predictable ways. This predictability forms the basis for selecting plants that support specific functions while maintaining balance within the body. As this understanding continues to develop, it reinforces the connection between plant chemistry and biological systems. Every compound plays a role in influencing how the body responds to internal and external conditions. The relationship ensures that plant-based compounds can be used in a controlled and reliable way when selection, preparation, and application remain consistent. The way plant material is prepared determines how its compounds are released and how effectively they interact with the body. Each preparation method influences how compounds move from plant tissue into a form that can be absorbed and used by biological systems. The process directly affects the strength, speed, and duration of the resulting physiological effect. When plant material is placed in water, soluble compounds dissolve and become available for absorption through the digestive system. Heat increases molecular movement, allowing these compounds to separate from plant structures more efficiently. This results in a preparation where compounds are evenly distributed within the liquid, creating a consistent and measurable effect when consumed. Some compounds require longer exposure to water to be fully released from dense plant structures, such as roots and bark. In these cases, extended heating allows deeper extraction of compounds that influence internal systems. This increases the concentration of active compounds within the preparation, producing a stronger effect when introduced into the body. Alcohol-based preparation extracts a wider range of compounds, including those that do not dissolve easily in water. This method preserves chemical structure and prevents rapid degradation over time. As a result, a stable and concentrated preparation that delivers a higher amount of active compounds in smaller quantities. Grinding or crushing plant material increases surface area, allowing compounds to be released more efficiently during extraction. This mechanical process breaks down plant structures and exposes internal tissues where active compounds are stored. The increased exposure improves the consistency and strength of the preparation by allowing more compounds to be released. Drying plant material reduces moisture content and slows chemical breakdown within plant tissues. This preservation method maintains compound stability and allows plant material to be stored for extended periods without significant loss of effectiveness. Proper drying ensures that compounds remain intact until they are extracted and used. When plant compounds are introduced into the body through ingestion, they pass through the digestive system before entering the bloodstream. Absorption occurs through the intestinal lining, where compounds move into circulation and are transported to target tissues. The efficiency of this process determines how quickly and strongly the compounds produce their effect. Topical application delivers plant compounds directly to the surface of the skin, allowing interaction with local tissue without entering systemic circulation. These compounds influence cellular repair and microbial balance within the applied area. The effect remains concentrated at the point of application, producing a controlled and localized response. Inhalation introduces volatile compounds through the respiratory system, allowing rapid interaction with neural pathways. These compounds travel through receptors linked to smell and influence brain activity associated with stress and relaxation. Because this pathway connects directly to neural systems, the response can occur quickly and with noticeable effect. Each method of delivery influences how plant compounds interact with the body and how long their effects remain active. Some methods produce rapid effects that diminish quickly, while others create a slower onset with longer duration. The choice of preparation determines how the compound is experienced and how it influences biological systems. The interaction between preparation method and biological response forms a controlled system where outcomes can be predicted based on process. Each step, from plant selection to final application, contributes to the measurable effect produced within the body. This enables plant-based compounds to be used in a structured and reliable way. Once plant compounds enter the body, their effects depend on how they interact with specific biological systems that regulate function and response. These systems include the nervous system, immune system, and circulatory system, each responding to compounds through defined mechanisms. The outcome of these interactions can be observed through measurable changes such as reduced pain, improved defense against infection, and increased blood flow. Within the nervous system, certain plant compounds bind to receptors that influence how signals are transmitted between cells. The interaction can reduce the intensity of pain signals or alter neural activity associated with stress response. As a result, a measurable change in how the body perceives and responds to internal and external stimuli. Some compounds influence neurotransmitter levels, which regulate mood, alertness, and sleep readiness. By increasing or decreasing these signals, plant compounds can shift the balance of neural activity. This produces observable changes in relaxation, focus, and the ability to transition into sleep. The immune system responds to plant compounds through pathways that regulate inflammation and cellular defense. Certain compounds reduce inflammatory signaling by limiting the release of molecules that trigger swelling and irritation. This supports tissue recovery and reduces the impact of prolonged inflammatory response. Other compounds stimulate immune cells, increasing their ability to identify and respond to foreign organisms. This increased activity strengthens the body's defense against bacterial and viral presence. As a result, a more efficient immune response during the early stages of infection. Within the circulatory system, plant compounds can influence blood vessel behavior and the movement of blood through tissues. Some compounds relax vessel walls, increasing blood flow and improving oxygen delivery to cells. This supports tissue function and can be observed through improved responsiveness and reduced strain. Other compounds affect how the heart functions by influencing contraction, strength, and rhythm. These interactions improve circulation efficiency and support stable blood flow throughout the body. As a result, a measurable improvement in how oxygen and nutrients are delivered to tissues. Plant compounds can also influence digestive processes by interacting with enzymes and muscle activity within the gastrointestinal tract. Some compounds stimulate digestive secretions, improving the breakdown and absorption of nutrients. This effect can be observed through reduced discomfort and improved efficiency in digestion. Other compounds relax smooth muscle within the digestive system, reducing pressure and improving movement through the intestines. This supports regular function and reduces buildup that leads to discomfort. As a result, improved digestive flow and a more stable internal environment. Each of these systems responds to plant compounds through direct interaction with receptors, enzymes, and cellular pathways. The specificity of these interactions determines the type and strength of the effect produced within the body. This enables plant-based compounds to influence multiple systems while maintaining predictable outcomes. The combined response of these systems creates a coordinated effect that supports overall function and stability within the body. Every compound contributes to this response based on its chemical structure and compatibility with biological processes. The interaction forms a connected system where plant compounds influence human physiology through measurable and repeatable mechanisms. Plant compounds can be grouped into categories based on their chemical structure and the type of effect they produce within the body. These categories provide a framework for understanding how different compounds interact with biological systems. Each category contains compounds that share similar properties and influence similar pathways. Alkaloids interact strongly with the nervous system by binding to receptors that regulate signaling between cells. These interactions influence pain perception, alertness, and response to stress. The effect can be observed through changes in sensation and neural activity. Flavonoids and polyphenols support cellular protection by reducing oxidative stress and limiting damage caused by reactive molecules. These compounds influence pathways that regulate inflammation and cellular stability. As a result, improved resilience within tissues and reduced impact of environmental stress. Terpenes influence microbial activity and neural response through their interaction with both cellular systems and sensory pathways. These compounds can disrupt microbial growth while also affecting mood and perception through their aromatic properties. This dual effect allows them to influence both physical and neural responses. Glycosides release active substances when broken down during digestion, allowing them to influence systems such as the heart and circulation. These compounds remain inactive until processed within the body, providing a controlled release of their active components. This forms a measured and sustained effect within target systems. Understanding these categories allows plant-based compounds to be selected with greater precision and used in a consistent and structured way. Every compound can be matched to a specific biological effect based on its structure and behavior. This framework supports a more controlled and predictable use of plant-based preparations. This structured understanding of preparation, delivery, and biological interaction completes the foundation of how plant compounds influence the body. Each step, from extraction to system interaction, contributes to a measurable and repeatable outcome. This consistency allows plant-based compounds to be used with confidence, producing reliable effects that support stability and function within the body. Plant compounds do not act in isolation when introduced into the body, and their effects can change when multiple compounds are present at the same time. The interaction creates a system where the combined influence of compounds can produce results that differ from the effect of each compound alone. Understanding how these interactions occur is essential for maintaining balance and consistency in plant-based use. When two or more plant compounds interact within the same biological pathway, they can increase or decrease the overall effect depending on how they influence that pathway. If both compounds act in a similar way, their combined effect may become stronger than intended. This can lead to an amplified response that exceeds the desired level of influence on the system. In contrast, some compounds can moderate each other's effects, creating a more balanced outcome. One compound may reduce the intensity of another while still allowing its primary function to occur. The interaction can prevent excessive stimulation while maintaining the intended physiological response. These interactions are not random, but follow predictable patterns based on how compounds bind to receptors and interact with enzymes. Every compound has a defined way of influencing biological systems, and when combined, these influences interact in a structured manner. This enables combinations to be adjusted based on how they behave within the body. When plant compounds influence different systems at the same time, they can produce a coordinated effect that supports multiple functions. One compound may act on the nervous system, while another influences circulation, creating a combined response that supports both relaxation and improved blood flow. This layered interaction allows plant-based preparations to address more than one condition simultaneously. The effectiveness of these combinations depends on how well the compounds complement each other. If the compounds support different aspects of the same function, they can enhance the overall outcome. If they interfere with each other, the result may be reduced effectiveness or unpredictable response. The timing of when compounds are introduced into the body also affects how they interact. Some compounds act quickly and produce immediate effects, while others take longer to build within the system. When used together, the difference in timing can influence how the combined effect is experienced. For example, a fast-acting compound may produce an immediate change, while a slower compound maintains that effect over a longer period. This forms a layered response where the initial effect is sustained rather than fading quickly. This type of interaction can improve consistency in how the body responds over time. The body's ability to process multiple compounds also influences how these interactions occur. Compounds are absorbed, distributed, and metabolized through shared pathways, and the presence of one compound can affect how another is processed. This can alter the strength and duration of the effect produced. Some compounds influence enzyme activity within metabolic pathways, affecting how quickly other compounds are broken down. If a compound slows this process, it can increase the duration of another compound's effect. If it accelerates the process, it can reduce the time that the compound remains active within the body. The interaction between compounds and metabolic processes creates a system where the overall effect depends on how compounds are processed, rather than just how they act individually. This adds another layer of complexity to plant-based use, requiring careful observation to maintain consistency. The selection of plant combinations must consider how compounds interact at multiple levels, including receptor binding, metabolic processing, and system response. Each of these factors contributes to the final outcome, and changes in one area can influence the overall effect. Some plant combinations are designed to enhance absorption, allowing compounds to enter the bloodstream more efficiently. Certain compounds increase permeability within the digestive system, improving how other compounds are absorbed. The interaction increases the effectiveness of the preparation without increasing the amount used. Other combinations are used to stabilize compounds, preventing rapid breakdown and extending their activity within the body. This enables the effect to last longer, reducing the need for repeated use. These stabilizing interactions contribute to a more consistent and sustained response. The use of plant combinations requires attention to balance, where the goal is to achieve a controlled and predictable effect. Every compound must be selected based on how it contributes to the overall outcome without disrupting the stability of the system. This supports that the body responds in a way that remains within a manageable range. In some cases, combining too many compounds can create complexity that reduces predictability. When multiple compounds interact across different systems, it becomes more difficult to anticipate how the body will respond. This highlights the importance of maintaining simplicity in combinations where possible. The body's response to combined compounds can also change over time as systems adapt to repeated exposure. This adaptation can alter sensitivity and affect how compounds interact with each other. Adjustments may be required to maintain the same level of effect over time. Understanding these interactions allows plant-based compounds to be used in a more controlled and structured way. By observing how combinations behave within the body, it becomes possible to refine their use and improve consistency. The process reflects the continued development of herbal practice as a system based on measurable outcomes. The relationship between compound interaction and biological response forms an essential part of how plant-based preparations are used. Each interaction contributes to the final effect, and the combined influence of multiple compounds creates a response that can be observed, repeated, and adjusted as needed. This structured approach ensures that plant-based compounds remain predictable and effective when used in combination. By maintaining awareness of how compounds interact, it becomes possible to use them in a way that supports stability and function within the body. This level of control allows herbal practice to operate with precision while remaining grounded in direct biological interaction. The timing of how plant compounds are used plays an important role in determining how they affect the body. Biological systems operate in cycles that change throughout the day, influencing how compounds are absorbed, processed, and responded to. These cycles affect functions such as digestion, circulation, and neural activity, creating variation in how the same compound behaves at different times. Some plant compounds produce stronger effects when used during periods of increased activity within the body. During these periods, systems such as circulation and metabolism operate at a higher level, allowing compounds to move more efficiently through the bloodstream. This increased movement can enhance the speed and intensity of the effect produced. In contrast, certain compounds are more effective when used during periods of rest, when the body shifts toward recovery and repair. During these times, neural activity decreases, and systems associated with relaxation become more active. Compounds that support these processes can produce a more noticeable effect when aligned with this natural state. The digestive system also follows patterns that influence how compounds are absorbed. When digestion is active, compounds introduced through ingestion are processed more efficiently and enter the bloodstream at a steady rate. If digestion is slowed, absorption may be delayed, altering the timing of the effect. The relationship between timing and absorption means that the same preparation can produce different results depending on when it is used. A compound taken during active digestion may produce a quicker and more consistent response, while the same compound used during reduced digestive activity may have a delayed or diminished effect. Circulation also varies throughout the day, affecting how compounds are distributed within the body. Increased blood flow allows compounds to reach target tissues more quickly, producing a faster response. Reduced circulation slows this process, extending the time required for the effect to become noticeable. Neural activity changes in response to environmental and internal factors, influencing how compounds interact with the nervous system. During periods of heightened alertness, compounds that reduce neural signaling may need to be stronger to produce a noticeable effect. During periods of reduced activity, smaller amounts may be sufficient to create the same response. The interaction between plant compounds and these biological cycles creates a system where timing can be adjusted to improve effectiveness. By aligning compound use with the natural state of the body, it becomes possible to produce more consistent and predictable outcomes. Repeated use of plant compounds can also influence how timing affects the body's response. As systems adapt to regular exposure, the timing of use may need to be adjusted to maintain effectiveness. This adaptation reflects the body's ability to regulate its response to ongoing stimulation. The duration of a compound's effect also plays a role in determining how often it should be used. Some compounds produce short-term effects that require repeated application to maintain their influence. Others produce longer-lasting effects that remain active within the body for extended periods. Understanding the duration of these effects allows for more accurate scheduling of use, ensuring that the compound remains active without excessive overlap. This prevents fluctuations in response that could lead to inconsistency in how the body reacts. The interaction between timing and duration creates a pattern where compounds can be used in a controlled and structured way. Each use contributes to a consistent level of activity within the body, maintaining stability over time. External factors such as physical activity, stress levels, and environmental conditions can also influence how timing affects plant-compound interaction. Increased physical activity may accelerate circulation and metabolism, altering how quickly compounds are processed. Stress can influence neural pathways, changing how compounds affect the nervous system. These factors must be considered when determining how and when plant compounds are used, as they can modify the expected response. Adjusting timing based on these conditions allows for greater control over the outcome. The coordination of timing, dosage, and preparation creates a system where plant compounds can be used with precision. Each factor contributes to the final effect, and adjusting one factor can influence the overall response within the body. Consistency in timing also supports the development of stable responses over repeated use. When compounds are used at the same time under similar conditions, the body's response becomes more predictable. This reduces variability and improves the reliability of the outcome. The use of plant compounds in alignment with biological cycles reflects a deeper understanding of how the body functions over time. By working with these natural patterns, plant-based preparations can produce effects that are both effective and consistent. This approach ensures that plant compounds are used in a way that supports the body's existing processes rather than conflicting with them. As a result, a more balanced interaction that maintains stability while producing measurable physiological effects. As this understanding develops, it allows for more refined control over how plant compounds are used within different contexts. Each decision regarding timing contributes to the overall effectiveness of the preparation, reinforcing the structured nature of herbal practice. The relationship between timing and biological response highlights the importance of using plant compounds within a framework that considers how the body changes over time. This awareness allows for more accurate application and improves the consistency of outcomes. By integrating timing into the overall system of plant selection, preparation, and dosage, it becomes possible to create a coordinated approach where each factor supports the others. This coordination ensures that plant compounds produce predictable effects while maintaining balance within the body. This structured use of timing completes another layer of control within herbal practice, allowing plant-based compounds to be used with greater precision. Each adjustment contributes to a measurable outcome, reinforcing the connection between plant chemistry and biological function. The response of the body to plant compounds is influenced not only by the compounds themselves, but also by the condition of the biological systems they interact with. Each system operates within a range of stability, and the effectiveness of plant compounds depends on how well those systems are functioning at the time of use. This means that the same compound can produce different effects depending on the state of the body. When biological systems are functioning within a stable range, plant compounds tend to produce predictable and consistent effects. The interaction between compound and system follows a defined pathway, allowing the expected response to occur with minimal variation. This forms a reliable outcome that can be observed and repeated under similar conditions. If a system is under stress or operating outside its normal range, the response to plant compounds may change. In some cases, the effect may be reduced because the system is less responsive. In other cases, the effect may be increased due to heightened sensitivity within that system. This variation highlights the importance of considering the condition of the body when using plant-based preparations. The nervous system plays a central role in regulating how the body responds to internal and external stimuli. Plant compounds that interact with neural pathways can influence perception, relaxation, and response to stress. The effectiveness of these compounds depends on how balanced the nervous system is at the time of use. When neural activity is elevated, compounds that reduce signaling may produce a noticeable calming effect. If neural activity is already low, the same compounds may produce a more subtle response. This variation reflects how the state of the system influences the outcome of the interaction. The immune system also affects how plant compounds produce their effects. Compounds that support immune response rely on the system's ability to recognize and respond to foreign organisms. If the immune system is functioning efficiently, these compounds can enhance its activity. If the system is compromised, the response may be slower or less effective. Inflammation plays a role in how plant compounds interact with tissues, as many compounds influence inflammatory pathways. When inflammation is present at moderate levels, compounds that reduce signaling can support recovery. If inflammation is excessive or prolonged, the same compounds may require more time to produce a noticeable effect. The circulatory system determines how quickly plant compounds are delivered to target tissues. Efficient circulation allows compounds to move through the body and reach areas where they are needed. If circulation is reduced, the distribution of compounds may be slower, affecting the timing and strength of the response. Digestive function also influences how compounds are absorbed and processed. Efficient digestion allows compounds to enter the bloodstream at a consistent rate, producing a stable effect. If digestion is impaired, absorption may be irregular, leading to variation in how the compound affects the body. The interaction between plant compounds and biological systems creates a dynamic relationship where both factors influence the outcome. The compound provides the potential for change, while the condition of the system determines how that change occurs. The relationship requires observation to ensure that the expected effect is achieved. Individual variation further influences how systems respond to plant compounds. Differences in genetics, lifestyle, and overall health contribute to how each person experiences the same preparation. This variation requires adjustments in dosage, timing, and selection to maintain consistency in results. Some individuals may respond quickly to small amounts of a compound, while others may require higher amounts to achieve the same effect. This difference is influenced by how systems process and respond to the compound. Recognizing this variation allows for more precise use of plant-based preparations. Adaptation also affects how systems respond over time. Repeated exposure to a compound can lead to changes in sensitivity, altering how strongly the system reacts. This may require adjustments in dosage or timing to maintain the desired effect. The balance between system condition and compound interaction creates a framework where plant-based compounds can be used in a controlled way. Each factor contributes to the overall response, and understanding this relationship allows for more consistent outcomes. The use of plant compounds requires attention to how systems change in response to internal and external conditions. Factors such as stress, physical activity, and environmental changes can influence system behavior, altering how compounds interact with the body. Adjusting for these factors improves the reliability of the effect. This approach ensures that plant-based compounds are used in a way that remains aligned with the body's current state. By matching the compound to the condition of the system, it becomes possible to produce a more accurate and predictable response. The relationship between plant compounds and biological systems reflects a balance between influence and response. The compound introduces a change, while the system determines how that change is expressed. This balance allows for controlled interaction that supports stability within the body. As this understanding develops, it provides a foundation for using plant-based compounds with greater precision. Each decision regarding selection, dosage, and timing can be adjusted based on the condition of the system, ensuring that the outcome remains consistent. This level of control reinforces the structured nature of herbal practice, where plant compounds are used in a way that reflects both their properties and the condition of the body. By maintaining this balance, plant-based preparations can produce reliable effects that support overall function and stability. Plant-based compounds operate within defined biological limits, and their effects are shaped by how they interact with existing systems rather than replacing them. Every compound supports or influences processes that are already present within the body, allowing those processes to function more efficiently or with greater stability. This means that plant compounds do not force change, but instead guide the body toward a more balanced state. These limits reflect how plant compounds interact with biological pathways through gradual and controlled mechanisms. Unlike highly concentrated substances that produce rapid and direct effects, plant-based compounds often act through multiple pathways at lower intensity. This forms a steady influence that builds over time rather than producing immediate and dramatic change. Some conditions require rapid and highly controlled intervention that exceeds the capability of most plant-based preparations. In these situations, the slower onset and lower concentration of plant compounds may not produce the necessary level of response. This does not indicate that the compounds are ineffective, but rather that they operate within a range suited to gradual adjustment rather than immediate correction. Plant compounds often support the body by influencing existing biological processes rather than overriding them. This approach allows systems to adjust naturally, reducing strain and maintaining stability. As a result, a gradual shift in function that becomes more noticeable over repeated use. This gradual influence can be observed in processes such as inflammation, digestion, and recovery from minor physical strain. Plant compounds may reduce inflammatory signaling over time, improve digestive efficiency through repeated interaction, and support tissue repair through consistent application. These effects build through cumulative influence rather than single use. Certain plant compounds are effective for mild to moderate conditions where the body can respond to support and adjustment. These include situations involving temporary discomfort, minor inflammation, and short-term disruption in normal function. In these cases, plant compounds can produce measurable improvement by working with the body's natural processes. In contrast, conditions involving significant structural damage or rapidly progressing infection may require intervention that acts faster and with greater precision. In these situations, plant compounds may provide supportive effects, but may not be sufficient as the primary method of response. This distinction highlights the importance of matching the strength of the intervention to the condition being addressed. The concentration of active compounds in plant preparations also affects the strength of their effect compared to more refined substances. Plant material contains a range of compounds that work together, often moderating the intensity of any single effect. This forms a balanced interaction that reduces the likelihood of excessive response while maintaining effectiveness within its range. This balance contributes to the stability of plant-based compounds, allowing them to influence the body without overwhelming biological systems. The combined action of multiple compounds creates a controlled effect that supports regulation rather than disruption. This is a key feature of how plant-based preparations operate within their limits. Variability in plant composition can also affect how consistently these limits are maintained. Differences in growing conditions, harvesting methods, and preparation can influence the concentration of active compounds. This variation can change how strongly a preparation affects the body, requiring careful selection to maintain consistency. Plant compounds do not act in isolation within the body, and their effects depend on the condition of the systems they interact with. If a system is functioning within a stable range, the compound can produce a predictable effect. If the system is disrupted, the response may be altered or reduced. The role of plant-based compounds is often to support regulation, recovery, and balance, rather than to provide complete correction of complex conditions. This role reflects how these compounds interact with existing biological pathways rather than replacing them. The effect is a contribution to overall function rather than a complete solution. Understanding these limits allows plant compounds to be used more effectively by aligning their use with conditions where they produce reliable outcomes. When used within their appropriate range, these compounds can provide consistent and measurable support to biological systems. This alignment ensures that their effects remain stable and predictable. The effectiveness of herbal practice depends on recognizing both the capabilities and the boundaries of plant compounds within the body. Every compound has a defined range of influence, and using it within that range allows for controlled and consistent outcomes. This awareness prevents overextension of plant-based methods into areas where they are less effective. The relationship between plant compounds and biological limits creates a framework where use is guided by both potential and constraint. Each decision regarding selection, preparation, and dosage must consider how the compound operates within this range. This supports that the effect produced remains within a manageable and beneficial level. By maintaining awareness of these limits, plant-based compounds can be used in a way that supports stability without creating imbalance. This approach reinforces the importance of precision and observation in herbal practice. Each application becomes part of a controlled system where outcomes can be predicted and adjusted as needed. This structured understanding of limits completes another layer of control within herbal practice, ensuring that plant-based compounds are used in a way that reflects both their strengths and their boundaries. By working within these limits, it becomes possible to produce consistent and reliable effects that support the body's natural processes while maintaining overall balance. The effectiveness of plant-based compounds depends on maintaining consistency in how they are selected, prepared, and used. Each step in this process contributes to the final outcome, and variation at any stage can alter how the compounds interact with the body. This requires a structured approach where each factor is controlled to produce predictable results. Consistency begins with plant selection, where the quality and condition of the plant determine the concentration of active compounds. Plants grown under stable conditions with sufficient nutrients and proper environmental support produce more reliable chemical profiles. Selecting plant material with consistent characteristics ensures that the starting point of the preparation remains stable. Preparation methods must also remain consistent to preserve the balance of compounds within the plant. Variations in temperature, time, and processing can change how compounds are released and how they behave during extraction. Maintaining the same preparation method allows the compounds to be delivered in a predictable form, ensuring that their effects remain stable across repeated use. Dosage plays a critical role in maintaining consistency, as the amount of compound introduced into the body determines the strength of the response. A controlled dosage allows the compound to interact with biological systems within a defined range, producing a measurable effect without causing imbalance. Adjusting dosage based on observation ensures that the effect remains consistent over time. The timing of use also contributes to consistency by aligning compound interaction with the body's biological cycles. Using plant compounds at similar times under similar conditions allows the body to respond in a predictable way. This reduces variability and supports the development of stable outcomes across repeated use. The interaction between these factors creates a system where consistency can be maintained through careful control and observation. Each factor supports the others, forming a structured process that produces reliable effects. When one factor changes, it can influence the entire system, highlighting the importance of maintaining balance across all stages. Repetition reinforces consistency by allowing patterns to be observed and confirmed over time. When the same preparation produces the same result under similar conditions, it becomes part of established practice. This repetition allows for refinement, where small adjustments improve accuracy and stability in the outcome. The body also contributes to consistency through its ability to respond in predictable ways when conditions remain stable. Biological systems follow patterns that can be observed and anticipated, allowing plant compounds to be used in alignment with these patterns. The interaction creates a coordinated response that supports stability within the body. Variability can occur when external factors influence how plant compounds are used or how the body responds. Changes in environment, physical condition, or stress levels can alter how compounds interact with biological systems. Recognizing these changes allows for adjustments that maintain consistency in the overall effect. The ability to adjust while maintaining structure is a key aspect of effective herbal practice. While consistency is essential, flexibility allows for adaptation when conditions change. This balance between stability and adjustment ensures that plant compounds remain effective under varying circumstances. The relationship between plant compounds and biological systems relies on maintaining a stable interaction over time. When this interaction is consistent, the body can respond in a predictable way, allowing outcomes to be measured and repeated. This predictability forms the basis for using plant-based compounds with confidence. The process of maintaining consistency extends beyond individual use and contributes to the broader development of herbal practice. As consistent results are observed across different individuals and conditions, knowledge becomes more structured and reliable. This enables plant-based methods to be refined and applied with greater precision. Each stage of the process, from plant selection to final use, contributes to the overall consistency of the outcome. By controlling these stages, it becomes possible to produce effects that are stable and repeatable. This level of control ensures that plant-based compounds remain effective within their defined range. The integration of consistency into herbal practice creates a system where plant compounds can be used in a controlled and predictable manner. This system supports the development of reliable methods that can be applied across different contexts while maintaining the same level of effectiveness. As this structure becomes more refined, it allows for greater confidence in how plant compounds are used. Each decision is guided by observation and supported by repeated outcomes, ensuring that the process remains grounded in measurable results. This approach reinforces the connection between plant chemistry and biological response. The continued emphasis on consistency ensures that plant-based compounds can be used as part of a structured system rather than as isolated applications. This system allows for controlled interaction between plant compounds and the body, producing effects that are both stable and measurable. By maintaining this level of consistency, plant-based compounds can be used in a way that supports long-term stability within the body. Each application contributes to a predictable outcome, reinforcing the reliability of herbal practice as a method based on structured observation and controlled use. The use of plant-based compounds requires an understanding of how different factors combine to influence the final outcome within the body. These factors include preparation, dosage, timing, system condition, and interaction with other compounds. Each factor contributes to how the compound behaves, and together they form a system that determines the overall effect. When these factors are aligned, plant compounds produce consistent and measurable results that support biological function. The interaction between compound and system follows a predictable pattern, allowing the effect to be observed and repeated under similar conditions. This alignment creates stability within the body by reinforcing existing processes rather than disrupting them. If one or more of these factors is not properly controlled, the outcome may vary from what is expected. A change in dosage, for example, can increase or decrease the intensity of the effect. A change in preparation method can alter how compounds are released and absorbed, affecting both timing and strength. These variations highlight the importance of maintaining control across all stages of use. The relationship between preparation and absorption plays a key role in determining how compounds enter the body. Proper extraction ensures that compounds are available in a form that can be absorbed efficiently through the digestive system or other delivery methods. If compounds are not fully released during preparation, their ability to interact with biological systems may be reduced. Dosage determines how strongly compounds interact with receptors and enzymes within the body. A controlled dosage produces a defined effect that remains within a stable range. If the dosage is too low, the effect may be difficult to observe. Timing influences how compounds interact with biological cycles, affecting both absorption and system response. Using plant compounds at consistent times allows the body to respond in a predictable way, improving the stability of the effect. Variations in timing can change how quickly compounds are absorbed and how strongly they influence specific systems. The condition of the body also affects how plant compounds produce their effects. Systems that are functioning within a stable range tend to respond more predictably, while systems under stress or imbalance may produce variable responses. This requires observation and adjustment to ensure that the intended effect is achieved. Interactions between compounds add another layer of complexity to how plant-based preparations function. When multiple compounds are present, they may enhance or moderate each other's effects, depending on how they influence the same biological pathways. Understanding these interactions allows for more precise use of combinations. The integration of these factors creates a system where plant compounds can be used with control and consistency. Each factor must be considered in relation to the others, ensuring that the overall effect remains stable. This structured approach allows plant-based compounds to produce reliable outcomes when applied correctly. Observation plays a central role in maintaining this system, as it allows for adjustments based on how the body responds. Each use provides information that can be used to refine future application, improving accuracy over time. The process ensures that plant-based compounds remain aligned with the body's needs. The ability to adjust while maintaining structure is essential for achieving consistent results. Small changes in one factor can be balanced by adjustments in another, allowing the overall effect to remain stable. This flexibility supports long-term use by accommodating variation without losing control. The body's response to plant compounds is influenced by both internal and external conditions, including stress, activity level, and environmental factors. These conditions can alter how compounds are absorbed and processed, affecting the final outcome. Recognizing these influences allows for more accurate application and improved consistency. The interaction between plant compounds and biological systems reflects a balance between influence and response. Every compound introduces a change, while the body determines how that change is expressed. This balance allows for controlled interaction that supports stability rather than disruption. Over time, the repeated use of plant compounds within this structured system leads to more predictable and refined outcomes. Patterns become clearer, and adjustments become more precise, allowing for greater control over how compounds influence the body. The process reflects the continued development of herbal practice as a system based on measurable results. The combination of preparation, dosage, timing, system condition, and interaction forms a framework that defines how plant-based compounds are used effectively. Each element contributes to the final outcome, and maintaining balance between them ensures that results remain consistent. This framework allows plant-based compounds to be used with confidence, as each factor can be adjusted to maintain stability within the body. The structured nature of this system supports reliable outcomes, reinforcing the connection between plant chemistry and biological response. As this understanding becomes more refined, it allows for greater precision in how plant-based compounds are applied. Each decision is guided by observation and supported by repeated outcomes, ensuring that the process remains grounded in measurable effects. The continued refinement of this system strengthens the reliability of herbal practice, allowing plant compounds to be used in a way that supports long-term stability. By maintaining control across all factors, it becomes possible to produce consistent and predictable results that align with the body's natural processes. Plant-based compounds continue to be used in modern contexts because their effects remain consistent when applied within structured systems. Despite advances in technology and changes in how substances are studied and produced, the underlying interaction between plant compounds and biological systems has not changed. These compounds still influence the body through the same pathways that were observed in earlier practices. Modern use of plant compounds often integrates both traditional observation and scientific analysis to improve accuracy and consistency. Traditional knowledge provides a record of how compounds behave under repeated use, while modern methods allow for detailed examination of their structure and function. This combination strengthens the ability to predict how plant-based compounds will influence the body. In contemporary settings, plant-based compounds are used to support functions such as digestion, circulation, immune response, and recovery from minor physical strain. These uses reflect the same patterns observed in earlier systems, where compounds were selected based on measurable effects. The consistency of these effects allows plant-based methods to remain relevant in modern practice. The availability of controlled growing environments has improved the consistency of plant material by reducing variation in environmental conditions. By regulating factors such as soil composition, light exposure, and water supply, it is possible to produce plant material with more stable chemical profiles. This reduces variability and improves predictability in how compounds affect the body. Modern preparation methods also contribute to increased consistency by allowing more precise control over extraction and concentration. Techniques that measure temperature, time, and compound release ensure that preparations contain a defined amount of active compounds. This level of control allows for more accurate dosage and improved reliability in outcomes. The ability to measure compound concentration provides a clearer understanding of how dosage influences effect. By knowing how much of a compound is present, it becomes possible to adjust the amount used to produce a specific response. This reduces uncertainty and allows plant-based compounds to be used with greater precision. Standardization has become an important aspect of modern plant-based use, where preparations are produced with consistent concentration and composition. This supports that each use produces a similar effect, reducing variability between different batches of the same preparation. Standardization supports the development of reliable methods that can be repeated across different contexts. Despite these advancements, the fundamental principle remains the same. Plant compounds influence the body through direct interaction with biological systems, and these interactions produce measurable effects. The consistency of this principle allows plant-based compounds to be used across both traditional and modern frameworks. Modern systems also emphasize the importance of safety and controlled use, recognizing that plant compounds must be applied within their defined range. By understanding how compounds interact with the body, it becomes possible to avoid excessive use and maintain balance within biological systems. This approach ensures that plant-based compounds are used responsibly and effectively. The integration of plant-based compounds into modern practice reflects a continued reliance on their ability to produce stable and predictable effects. While methods of analysis and preparation have evolved, the core interaction between compound and system remains unchanged. This continuity allows plant-based methods to adapt while maintaining their foundation. Research into plant compounds continues to expand, providing new insights into how they interact with biological systems. This research builds on existing knowledge, refining understanding of how compounds function at a detailed level. Each new finding contributes to a more precise application of plant-based compounds. The use of plant compounds in modern contexts often involves combining traditional knowledge with measured data to create a more structured approach. This combination allows for both flexibility and control, ensuring that plant-based methods remain adaptable while maintaining consistency in outcomes. The continued relevance of plant-based compounds is supported by their ability to operate within the body's natural processes. Rather than replacing these processes, they interact with them in a way that supports stability and function. This alignment allows plant-based compounds to be used effectively across different contexts. The role of plant compounds in modern use reflects a balance between tradition and refinement. Traditional practices provide a foundation of observed effects, while modern methods enhance precision and control. Together, these approaches create a system that supports consistent and measurable outcomes. The reliability of plant-based compounds in modern settings depends on maintaining the same principles that guided earlier use. Consistency in selection, preparation, dosage, and timing ensures that the compounds continue to produce predictable effects. This continuity reinforces the structured nature of herbal practice. As plant-based compounds continue to be studied and applied, their role within modern systems becomes more clearly defined. Every compound can be evaluated based on how it interacts with specific biological pathways, allowing for more targeted use. This precision supports the development of more effective and controlled applications. The integration of plant compounds into modern practice demonstrates that their effectiveness is not limited to historical use. Their continued application reflects a stable and reliable interaction with biological systems that remains consistent across different contexts. This consistency ensures that plant-based compounds remain a relevant and valuable part of how the body can be supported. The combination of traditional knowledge and modern refinement allows plant-based compounds to be used in a way that is both structured and adaptable. Each use contributes to a system where outcomes are predictable and measurable, reinforcing the connection between plant chemistry and biological response. This ongoing development ensures that plant-based compounds continue to operate within a controlled framework where their effects can be observed, adjusted, and applied with precision. By maintaining this structure, plant-based methods remain effective in supporting stability and function within the body. The way plant compounds are delivered into the body plays a critical role in determining how quickly they act and which systems they influence. Different methods of delivery change how compounds are absorbed, how they move through the body, and how long their effects remain active. This forms variation in both the speed and location of the response. When plant compounds are consumed through ingestion, they pass through the digestive system before entering the bloodstream. The process involves breakdown, absorption through the intestinal lining, and transport through circulation to target tissues. Because this pathway requires multiple steps, the onset of effects is gradual and develops over time. This slower process allows compounds to be distributed throughout the body, influencing multiple systems simultaneously. The effect tends to be more sustained as compounds are released and absorbed over a longer period. This makes ingestion suitable for producing steady and consistent changes in internal systems. In contrast, topical application delivers plant compounds directly to the surface of the skin, allowing them to interact with local tissue without passing through the digestive system. This method produces a more targeted effect, where compounds influence the specific area to which they are applied. The response is localized and does not rely on systemic distribution. Topical delivery allows compounds to support processes such as skin repair, moisture balance, and reduction of surface inflammation. Because the compounds remain concentrated in one area, the effect can be observed directly at the point of application. This forms a controlled interaction that limits influence to a specific region. Inhalation introduces volatile plant compounds through the respiratory system, allowing them to interact rapidly with neural pathways. These compounds travel through receptors associated with smell, which are directly connected to areas of the brain that regulate stress response and emotional processing. This connection allows inhaled compounds to produce effects quickly. Because inhalation bypasses the digestive process, the onset of effects can occur within a short period. This rapid interaction makes inhalation effective for influencing neural activity and producing noticeable changes in relaxation and alertness. The effect may be shorter in duration compared to ingestion, but it occurs more quickly. Each delivery method influences how plant compounds move through biological systems and how strongly they affect specific areas. Ingestion supports broader distribution, topical application targets localized areas, and inhalation influences neural pathways through rapid interaction. These differences allow plant compounds to be applied based on the desired outcome. The speed of absorption varies depending on the method used, affecting how quickly the body responds. Faster absorption produces immediate changes, while slower absorption creates a gradual effect that builds over time. Understanding this difference allows for more precise control over how plant compounds are used. The duration of effect is also influenced by how compounds are delivered. Compounds absorbed slowly tend to remain active for longer periods, while those absorbed quickly may diminish sooner. The relationship between speed and duration creates a balance that must be considered when selecting a delivery method. The ability to target specific systems within the body depends on how compounds are introduced. Some methods allow compounds to interact with multiple systems, while others focus on a single area. This targeting improves the efficiency of the compound by directing its effect where it is needed most. For example, compounds used to support digestion are often delivered through ingestion, allowing them to interact directly with the digestive system. Compounds used to influence skin condition are applied topically to produce a localized response. Compounds used to affect mood and relaxation may be inhaled to produce a rapid neural response. The choice of delivery method must align with the intended effect to ensure that the compound interacts with the correct system. Using the appropriate method increases the likelihood of producing a measurable and consistent outcome. This alignment reduces variability and improves the effectiveness of plant-based compounds. The concentration of compounds can also vary depending on how they are delivered. Some methods allow for higher concentration in a specific area, while others distribute compounds more evenly throughout the body. This difference influences both the intensity and range of the effect. Combining delivery methods can create a layered response where multiple systems are influenced at the same time. For example, ingestion may provide a sustained internal effect, while topical application targets a specific area. This combination allows for a more comprehensive interaction with the body. The interaction between delivery method and biological response creates a structured system where plant compounds can be used with precision. Each method offers a different pathway for compounds to enter the body and produce their effect. By selecting the appropriate method, it becomes possible to control how and where the compound acts. Understanding these differences allows plant-based compounds to be used in a way that maximizes their effectiveness while maintaining stability within the body. Each method contributes to a specific type of response, and choosing the correct one ensures that the outcome remains predictable. This structured approach to delivery completes another layer of control within herbal practice. By aligning method with desired effect, plant compounds can be applied in a way that supports both accuracy and consistency. This supports that their influence remains targeted, measurable, and reliable across repeated use. The effectiveness of plant-based compounds depends on how consistently their effects can be observed and repeated under similar conditions. This repeatability allows patterns to form, where specific compounds are linked to specific outcomes through direct experience. When these patterns remain stable, plant-based use becomes more predictable and easier to apply with accuracy. Each plant compound produces a measurable effect based on how it interacts with biological systems. These effects can be observed through changes in processes such as inflammation, digestion, circulation, and neural activity. When the same compound produces the same change across repeated use, it establishes a reliable connection between the compound and its outcome. This reliability allows plant-based compounds to be selected with greater confidence. Instead of relying on general assumptions, selection is based on known interactions that produce consistent results. This forms a structured approach where plant compounds are matched to specific conditions based on observable evidence. The ability to repeat results depends on maintaining consistency in preparation, dosage, and timing. If these factors remain stable, the compound interacts with the body in a predictable way. If they vary, the outcome may change, reducing the reliability of the response. This highlights the importance of controlling each stage of use. Observation plays a central role in confirming whether a compound is producing its intended effect. By monitoring how the body responds, it becomes possible to determine if the outcome matches expectations. The process allows adjustments to be made when necessary, improving accuracy over time. When outcomes are consistent, they reinforce the relationship between plant compounds and biological response. Each successful use strengthens confidence in the compound's effect and supports its continued application. The process builds a system where knowledge is based on repeated and measurable results. Variation can occur when conditions change, affecting how compounds interact with the body. Differences in environment, system condition, and individual response can influence the outcome. Recognizing these variations allows for adjustments that maintain consistency despite changing conditions. The ability to adapt while maintaining structure is essential for preserving repeatability. adjustments must be made carefully to ensure that the overall effect remains stable. This balance between consistency and flexibility allows plant-based compounds to remain effective across different situations. The use of plant compounds also involves recognizing patterns that extend beyond individual cases. When the same effect is observed across multiple individuals, it confirms that the compound produces a consistent biological response. This broader pattern strengthens the reliability of the compound and supports its use in a structured system. Over time, these patterns form the basis of established practice, where plant compounds are used with a clear understanding of their effects. This enables for more efficient selection and reduces uncertainty in how compounds will influence the body. Each application becomes part of a system grounded in observable results. The relationship between plant compounds and repeatable outcomes reflects a stable interaction between chemical structure and biological function. Every compound interacts with the body in a defined way, producing changes that can be measured and confirmed. This stability allows plant-based methods to remain consistent over time. The ability to predict outcomes improves as more observations are collected and patterns become clearer. With increased experience, the selection of plant compounds becomes more precise, and the likelihood of achieving the desired effect increases. This progression reflects the refinement of herbal practice through repeated use. Consistency in outcomes also supports the development of more structured methods, where plant compounds are used according to defined principles. These principles guide selection, preparation, and application, ensuring that results remain stable across different contexts. The integration of repeatability into plant-based use ensures that compounds can be applied with confidence. Each use reinforces the connection between compound and effect, allowing the system to become more reliable over time. This reliability is essential for maintaining consistency in outcomes. The ability to observe, repeat, and confirm results forms the foundation of how plant-based compounds are used effectively. Without this process, outcomes would remain uncertain and difficult to predict. By maintaining this structure, plant-based methods remain grounded in measurable biological interaction. As this system continues to develop, it allows for greater precision in how plant compounds are applied. Each observation contributes to a clearer understanding of how compounds influence the body, improving accuracy and control. This ongoing refinement ensures that plant-based compounds remain effective and reliable. The connection between repeatability and reliability ensures that plant-based compounds can be used within a structured framework where outcomes are predictable. Each factor, from preparation to application, contributes to maintaining this consistency. This framework supports the continued use of plant compounds in a way that remains stable and measurable. Through this process, plant-based compounds become part of a system where their effects are not only observed, but understood and anticipated. This level of understanding allows for more controlled and consistent use, reinforcing the connection between plant chemistry and biological response. This structured approach ensures that plant-based compounds can be used with confidence, producing effects that are both predictable and repeatable. By maintaining consistency and observing outcomes, it becomes possible to apply these compounds in a way that supports stability and function within the body. The use of plant-based compounds represents a system built on observation, consistency, and measurable interaction with the body. Every compound produces a defined effect based on how it interacts with biological systems, and these effects can be observed, repeated, and refined over time. The process has allowed plant-based methods to develop into a structured approach where outcomes remain predictable when conditions are controlled. The relationship between plant compounds and human physiology is based on direct interaction at the level of receptors, enzymes, and cellular pathways. These interactions influence processes such as inflammation, digestion, circulation, and neural activity. Because these processes operate within defined biological systems, the effects of plant compounds follow patterns that can be measured and confirmed. Each plant represents a complex chemical system shaped by environmental conditions and biological function. These compounds are produced as part of the plant's survival strategy, yet they continue to function when introduced into the human body. This connection allows plant compounds to influence human systems in ways that reflect their original biological purpose. The effectiveness of plant-based compounds depends on how well they are aligned with the body's existing processes. When used correctly, they support regulation, recovery, and stability by interacting with systems that are already present. This alignment ensures that their effects remain controlled and predictable rather than disruptive. The structured use of plant compounds requires attention to preparation, dosage, timing, and method of delivery. Each of these factors contributes to how compounds are absorbed, distributed, and processed within the body. By controlling these variables, it becomes possible to produce consistent outcomes across repeated use. The ability to predict outcomes improves as patterns become more clearly defined through repeated observation. Each use reinforces the connection between compound and response, allowing adjustments to be made with greater precision. The process ensures that plant-based compounds can be used in a way that remains accurate and reliable. Plant compounds operate within biological limits, supporting processes that the body is already capable of performing. They do not replace these processes, but influence them in a way that promotes balance and stability. This role defines how plant-based methods are used within their appropriate range. The continued use of plant-based compounds reflects their ability to produce measurable effects that remain consistent across different conditions. Despite variation in environment, preparation, and individual response, the underlying interaction between compound and system remains stable. This stability allows plant-based methods to remain effective over time. The integration of traditional observation with modern understanding has strengthened the ability to use plant compounds with greater precision. While methods of analysis have evolved, the core interaction between plant chemistry and biological response has remained unchanged. This continuity allows plant-based compounds to be applied within both traditional and modern frameworks. The use of plant compounds also requires an awareness of their limitations, ensuring that they are applied in situations where they can produce reliable outcomes. This understanding prevents misuse and supports the development of a balanced approach where plant-based methods are used appropriately. The combination of preparation, dosage, timing, delivery, and observation creates a complete system where plant compounds can be used with control and consistency. Each element contributes to the final outcome, forming a structured process that allows for predictable interaction with the body. As this system is applied over time, it becomes more refined, allowing for greater accuracy in how plant compounds are selected and used. Each observation contributes to a deeper understanding of how compounds influence biological systems, improving the ability to produce consistent results. The relationship between plant compounds and the body reflects a balance between chemical influence and biological response. This balance allows for controlled interaction that supports stability without overwhelming existing systems. As a result, a method of use that remains aligned with the body's natural function. The continued refinement of this approach ensures that plant-based compounds remain a reliable part of how biological systems can be supported. By maintaining structure and consistency, it becomes possible to produce outcomes that are both measurable and repeatable. This understanding represents the culmination of observation, refinement, and application across time. Each step in the process contributes to a system where plant compounds can be used with confidence, producing effects that remain stable and predictable. Through this structured approach, plant-based compounds continue to demonstrate their ability to influence the body in ways that are consistent with biological function. This consistency allows them to remain relevant and effective as part of a controlled system grounded in measurable interaction. The complete system of herbal practice reflects a connection between plant chemistry and human physiology that is both stable and adaptable. Every compound contributes to this system through its specific interaction with biological pathways, allowing for controlled and predictable outcomes. By maintaining attention to each factor involved in this process, it becomes possible to use plant-based compounds in a way that supports long-term stability within the body. This approach ensures that outcomes remain consistent, reinforcing the reliability of plant-based methods. This final integration of principles completes the framework of how plant compounds are used, connecting selection, preparation, and application into a unified system. Each part of this system contributes to a measurable effect, allowing plant-based compounds to be used with precision and confidence. Thank you for listening to the Sleepy Mind channel. I hope you enjoyed learning about herbal remedies.